Hello, everyone. Thank you for joining us for our webinar. We're really excited to have you with us today. I'm pleased to have two really experienced experts in remediation, Dr. Natalie Capiro and Dr. Kurt Pinnell, and it's really an honor to have both of you speaking today on our webinar series. So as a quick introduction, Natalie is an assistant professor in the Department of Civil and Environmental Engineering at Auburn University. And her research interests include environmental microbiology, bioremediation, fate, and transport of legacy and emerging contaminants in natural systems, and nanotechnology, biological interactions in the environment. And then there's Dr. Kurt Pinnell, who's the 250th anniversary professor of engineering at Brown University. Kurt's research addresses three main topics: soil and groundwater remediation, engineered nanomaterial fate and transport, and environmental toxicology. His current research focuses on in situ remediations of the PFAS and environmental exposure monitoring and metabolic responses, and the use of engineered nanomaterials for subsurface characterization. So during the webinar today, Kurt and Natalie will talk about something that probably doesn't get enough attention in research. Back diffusion is a common concern with remediation efforts, especially in low permeability areas. So I'm really delighted to have them on our series. Their talk today is going to focus on ways that you can enhance this process biologically to get greater long-term success. So many of you have attended our webinar series before and know this, but the recording is going to be available on the resource section of the Microbial Insights website. That will happen within the next week. And you don't have to contact us, and link will automatically be emailed to all registrants as soon as it's available. And at the end of the morning's webinar, there's going to be a question and answer period. So if you have questions during the presentation, go ahead and type those into that panel on your GoToWebinar software, and we'll answer them as quickly as we can. And then if you think about other questions after the webinar, feel free to reach out to us. We can always get with Kurt and Natalie and have them answer any additional questions. So you can email us at info@microbe.com with anything additional that you need. So without any further ado, Kurt, I'll go ahead and turn it over to you.
Okay, sounds good. Can you hear me okay?
Yes, you sound great.
All right. Welcome, everyone. Just a little bit of background of how we're going to set this up. I'm going to talk for about 20 minutes on primarily physical and chemical processes that influence mass transfer. And then Natalie is going to focus on the biological part of it because that's her expertise. So just so you have that general sense of how we're going to present. So we'll switch over after about 20 minutes to her set of slides. All right, so what I wanted to do is give a little bit of background about where we're coming from. And one of the things we're focused on now is how do we manage complex sites that have, and a lot of these are legacy sites that have been around for a while. And I was on an NRC panel that Mike Cavanaugh led. And we sort of looked at this issue and how we handle this. And so the first slide is the status of complex sites, not STIs, in the United States. And this is the good. We've eliminated a lot of the acute threats to human health. We have protective remedies in place at most sites. We've handled a lot of the underground storage tank sites and achieved no further action. And there's a number of NPL sites that have been delisted. And a lot of RCRA facilities have been handled. And so this is also a good thing. And there are a number of brownfield cleanups. So these are the positive things that have happened with a lot of these legacy sites that we're dealing with. And then the bad part of it is, and then the part we're facing now is the estimated cost to remediate. And from this study, the NRC, we estimated there are about over 125,000 sites. And the cost may be on the order of 127 billion. And now the ugly part is that we, in the last few years, we've realized we have to deal with PFAS. And a lot of our research and regulatory concerns are now related to this compound, which often occurs at sites that also have legacy contaminants such as BC and TCE. And we have to deal with those source zone contaminants. So one thing we sort of set up, this is a conceptual diagram about how can we address complex sites more effectively. And this is sort of the lead into the talk we're giving today. And on this graph, I just show groundwater concentration. This could be mass. And on the x-axis is remediation time or cost. And I have sort of three lines showing, what this might look like, how it decreases, how the concentration decreases as you apply a remedy. But we also often get to this point where we have a stalled remediation and we can't achieve the remediation target. And we may have addressed a lot of the source zone contamination, and then we can't really get down to groundwater limits or whatever the regulatory limit we're trying to reach. And this is where the back diffusion becomes important. How do we get past this stalled phase? So one of the things we're going to be focused on today is how do we develop an improved understanding of the source zone and down gradient plume behavior. So that's a lot of how the things we talk about today will relate. Another approach, and we won't really talk about this today, is combined remedies. So the idea here is we could combine a remedy in parallel, so that means at the same time, same location, to get more rapid, so we get a more rapid decrease in this curve. And the third thing, which is primarily what we're focused on here, is how do we combine remedies in series, sometimes referred to as a polishing step, to jumpstart a stalled site or transition to monitored natural attenuation. So, and a lot of times this is where the back diffusion and those types of processes come into play, because we have this slow release and we just can't get down to the concentrations we need to get to. So the idea is, how do we modify things, or how do we use processes over a longer periods of time to achieve this remediation target? Okay, I'm going to show you some of our prior work. We're going to start with source zones that have DNAPL in them and look at mass transfer from these types of sites, these types of locations, and then we're going to transition to looking at the plume. So we want to sort of have a comprehensive look here. And one way we did this before was to run these pretty large scale studies in the laboratory where we spilled DNAPL in a source zone, and then we could image it. And then we could look at the behavior over time. And we had a down gradient plume region, and these are little sampling ports, so we can actually take samples in time and space to look at how the plume is evolving. And so this gives us a good sense of how things change over time as we remove mass from the source zone. So what I want to show here is this is on the left is a picture with a backlit. We can see we dyed this is a PC DNAPL, we dyed it red, and then we can image this and actually get a distribution between ganglia and pools and have a sense of what the source zone looks like and characterize it that way. And these are the sampling ports. So we have these nice, they're what we call low dead volume. So we're not collecting a lot of water. One of the problems is when you collect the sample in this type of a domain, if you pull out a lot of sample, you actually influence the flow field, and you don't want to do that. So, and you also don't want sand to go into the port where you're trying to take a sample. So this is how we address that. And on this next slide, what I show here is changes in source zone architecture as we remove mass. So as we go down, we have initial saturation profile, and we actually use a surfactant flood to accelerate the removal of mass because it just accelerates dissolution. And so we have three different scenarios here. The initial profile after a first flood where we basically reduce the mass quite a bit, and then after a second flood we reduce it quite a bit more. And what I show you is three different spill scenarios. We call this one on the left high ganglia to pool ratio. Okay, so this means we have a lot of ganglia or small droplets. And then we had a mid ganglia to pool ratio where we had a sort of a mix of pools above low permeability lenses and then ganglia here. And then we had a mid to low ganglia to pool and then a low ganglia to pool. What this, the one on the right represents is mostly a pooled scenario. You can see this accumulation at the bottom of the cell. Quite a bit of the mass was in this pooled location. And what we wanted to do was see, as we remove mass, how this impacted the down gradient plume. And as you might know, when a lot of the mass transfer relates to the area that's available for that mass transfer to occur across. And when you have a lot of ganglia, you'll get much more mass transfer. In contrast, when you have pools, you have much lower surface area for the amount of mass spilled, and it's much harder to get the mass out. So on the left-hand side is probably the easier type of spill to remediate. And on the right-hand side is your worst case scenario where you have a lot of pooling. You may remove ganglia rapidly, but then you're left, if you look at the lower right, with these pools that persist and quite a bit of mass that's still at the site. So that's sort of our scenario we're looking at. We ran these different experiments. And what I want to show you now is some of the examples of the what the down gradient plume looked like. So this is for the high ganglia to pool ratio. So this is a lot of ganglia. And you can see the pictures on the left-hand side. Okay, so we have most of the mass recovered at the bottom, just a few pools, and you can see the down gradient plumes. So what this shows you here is that the down gradient plume is controlled by the location of the DNAPL, which is obviously very much influenced by the heterogeneity at the site. And as mass is removed, you basically lose all the smallest droplets or the highest surface area ganglia. And then you end up with these sort of stringers. The plume looks like stringers where you have mass coming off of these zones where there's still NAPL present. Okay, so this is sort of plume evolution in the source zone. And you can see how it changes over time. All right, so this slide, what I'm trying to show here is some of how we treat mass transfer. And what I have here is an upper left is a NAPL droplet, and we have the water flowing past it. We have a stationary or no flow boundary, so this is the boundary layer. And so we have a concentration that's very high, and then we have a boundary layer. And then we have the concentration out in the bulk aqueous phase. And in the upper right, what I show you is how we would treat this. A change in concentration over time is equal to an effective mass transfer coefficient times a linear driving force. Okay, and that driving force is basically the equilibrium concentration minus the actual concentration. So as you get closer and closer to equilibrium, you basically, the driving force goes down and your change in concentration with time decreases. Okay, so when these are very much separated, so your actual concentration is very much lower than the equilibrium, the driving force is greater, and you'll have greater mass transfer. One thing to note, this effective mass transfer coefficient, what that really includes is the actual mass transfer coefficient and the interfacial area. So if you try to model these systems, you need to know the interfacial area of NAPL, and it may change over time, and so you have to account for that. Also, you can relate this to the flow rate where little q is the Darcy velocity, and this k naught is if you have a flow interruption. So if you stop the flow, you can also describe the mass transfer. So when q goes to 0, then you just have this term on the right, which is mass transfer under a no flow condition, okay, which is just diffusional mass transfer. So we can put this in our transport equation. You may be familiar with this as just a simple advective dispersive transport equation. And what I have highlighted in here in red is on the left-hand side, what we want to be able to predict is our change in concentration with time. Here we have our dispersion term, our advective flow term, our absorption term, okay? And then we have our source term. So the NAPL is basically a positive sign because we're adding mass to the system. Absorption, for the most part, you're losing mass from the system, and Darcy velocity is taking mass away. But this is our source term, and so it's basically adding mass to the system. This is how we describe that mathematically. All right, I'm going to shift gears a little bit, and what I've showed you previously is how we conceptualize dissolution and how we might model that, and I'll do some modeling later. But a lot of times we use these compartment-based conceptual site models, which are very useful to get an idea of, we want to know where is the mass. So we can write all these equations and do predictions, but when you go to a field site, you want to sort of figure out where the mass is. And this is from work that Tom Sale and Chuck Newell did. And this is what they call their 14 compartment model. And this is an example of an early stage NAPL site and using aqueous equivalents. Okay, and this is what they came up with. Most of the mass is in the NAPL. So this red zone or red box is high concentration. Okay, and then these are lower, and you have some in the aqueous phase. So we have the source zone and the plume. So you can think of it almost like those figures I showed you before, the pictures I showed you of the experimental cell, where this is the source zone and then the plume, and over time we're moving mass into the plume. Now, they then sequence this. So this is, again, Tom's work and Chuck's work. And what I want to show you is early stage. You see the mass is in the DNAPL. You go to middle stage, everything's moderate, and everything's low. And sort of, I was never sure how they come up with these values. So, you know, you sort of can guess or use these equivalents to get at those. But we see there's not, like at late time, there's not much in the plume, but in at a lot of sites, when we go to late time, that's where all the mass is. And when we try to deal with back diffusion, we're in the plume zone, and that's where we're concerned. And we want to know how much is in the low permeability zone and how much is in the transmissive. So, so this is the area that we're really interested in terms of back diffusion, which may cause a site to persist for very long periods of time. So a different way to approach this is to use a contaminant phase distribution model. We actually calculate the mass in these different phases. And what I mean by that is what's in the water and what's in the soil and what's in the NAPL. And for the most part, we'll assume we don't really have much of a gas phase because we're in the saturated zone. And if you take a core, you can calculate, if you take a core sample, you can calculate how much is in the solid, water, gas, and NAPL phase based on these different concentrations. So there's a concentration in the water, the volumetric water content, concentration gas phase, volumetric gas content, bulk density, concentration of solid phase, and then concentration of NAPL and the volumetric NAPL content. What this allows you to do is add them all together. You can't just add together concentrations because they represent different phases, right? But if you express them all as milligrams per liter total, then you can add them together and you can then figure out what your contaminant mass distribution is. So I applied this approach to the 14 compartment model. And this is a slightly different site, but the same type of idea where we had early stage where we say, okay, 5% NAPL, and we can see all the mass is in the DNAPL. And this is very similar to what Tom Sale and Chuck Newell showed. And then at middle stage, we get moderate. We're losing mass from the source zone. We're starting to get mass in the absorb phase. Mass is starting to pick up. Right? And at the late stage, this is where it diverges. What you can see here, most of the mass is absorb phase in low permeability media. And this is the part we need to focus on. So this is where back diffusion really comes into play. We have low transmissive or low permeability media, a lot of absorb phase. That's where all the mass is. And that's really why the work we're doing with back diffusion and how biological processes influence, that's how it relates to what I'm talking about now. Okay, now what I'm going to show you, I'm going to shift gears again and show you some modeling. This was done by Linda Abriola's group. We work quite closely with Linda. Both Natalie and I do. We have a number of CERTA projects. And what I want to show you is another way to look at this is to take this information that I just presented about the dissolution and the absorption in the plume and treat this in a modeling context for a theoretical site. So this was generated, this is the source zone. Here's a permeability field. This is actually from a representation of a real site. And then down here, we see that this is there was a DNAPL spill. So they had a hypothetical spill. And then we have a transport model. So MT3D is used for that. And then post-processing, we looked at mass flux and how that, and I'll show you some of that data, those data. We had a number of different types of porous media. So these are just symbols for those, but just shows you the conductivity and the uniformity and the medium grain size. So the key thing here is we're coupling rate-limited nonlinear processes to describe contaminant fate and transport. So it's important to remember that many, not all models, but many models assume equilibrium, linear absorption, right? And part of our argument is that we need to account for rate limitations and nonlinear processes. And I'll show you some examples of how important these things can be in our predictions. So here's the initial condition, and here's a reference if you're interested. So this is the permeability field, slices through that. Here's the aqueous phase concentration, absorb phase concentration, and the NAPL saturation. So you can see this is in the source zone, and the mass will then be removed over time from the source zone, and we'll look at how the source zone changes over time. All right, this is at 4 years, and obviously we're coming, what I want to show you in this graph, this is time here in the upper right, aqueous concentration. Imagine that we're pumping from this site, and the key thing is we see this rapid reduction in mass which is typical for most sites. We're pulling out a lot of mass, this is also on a log scale, so it's even accentuated. But then we get to a flat part of the curve, and this is sort of what I was showing in that earlier graph. So how do we get off of this flat part of the curve? And you can see from this period from about 20 years to 62 years, where nothing much is happening, all right? And that's the problem we face for many of these sites. How do we deal with that? How do we get down to this last part? And we don't necessarily want to take 100 years to clean up the site. All right, here's at 62. So here we're on this next shelf, right? And what's happening here, we've now removed a lot of mass. And you can see there's no more NAPL mass really. We have a little bit of sorb mass and aqueous phase mass, okay? So we're still seeing some release from this site, and it's a little bit hard to see, but there's still sorb mass down here, and some, of course, at the top you can see more clearly. So we go through another period where we drop from about 1 milligram per liter. We have a pretty big drop here in this next period, and what we're doing here is pulling off a lot of this sorb mass now. And then we go, I just skipped a couple of these. We see these, again, these drops. And what happens here is we've cleaned up different phases, different parts of low permeability media where the absorption is. And that's why you get these sort of step-like functions. And now we want to focus at this low time. Really at the really late time, we have just sorb mass. We're just bleeding out slowly. You see the aqueous concentrations are low, but they continue on. And this, you can also think of, this is like a down gradient plume now, because we have no real NAPL mass left. The sorb mass is relatively low. But I blew up the right-hand corner, and what I want to show you is different representations of absorption. So the green line is no absorption, the blue line is linear absorption, then nonlinear absorption, and then rate-limited absorption. Okay, and you can see if there's rate-limited desorption, you can have a very long progression here. Okay, very long tailing. But the other thing interesting is that with no absorption and linear absorption, you get about the same. Okay, and this means that diffusion is back diffusion is dominating your system. Okay, so that some of the take-home points, total persistent time is obviously highly correlated with NAPL dissolution. But once we get rid of the NAPL, dissolved and absorbed phase mass storage can sustain these plumes for long periods of time. And here's what I was mentioning about back diffusion can play an important role. And you can see, this really stands out because if there's no absorption or linear absorption, you get kind of the same result, which means that it's influencing your system quite strongly. The last part is this late persistent can be strongly influenced by how you represent absorption, whether it's linear or nonlinear, and how the subsurface heterogeneity is represented in your modeling. I want to show you one last thing, the effect of isotherm type. So I mentioned that whether you use linear or nonlinear. This is an example of some absorption isotherms that are fit to data. I fit a linear model, Langmuir model, Freundlich model to these data. And we're just going to use this narrow part of the curve, the lower concentration part, where you may just say, oh, it's good enough to fit a linear isotherm. These data look pretty linear to me. These are the breakthrough curves that you would obtain if you ran an experiment, a pulse injection with this based on these isotherm data. So the nice symmetric is our equilibrium linear model, okay? And then we have Langmuir, you get self-sharpening and a long tail. And then we have Freundlich, you get delayed breakthrough and then long tailing. And the Freundlich is this dashed one. And what you can see here, it's actually exhibiting the strongest absorption, okay, in this low concentration zone. And that's why you get such delayed breakthrough and such long tailing. So tailing can occur just because of the isotherm type, not due to mass transfer limitations. This is equilibrium, okay? So you can have this kind of dramatic tailing even if you assume equilibrium, okay? So just some take-home messages. Compartment type conceptual models can be very useful, but if possible, it's best to calculate the contaminant mass using chemical and soil properties. And I strongly encourage, I go to a lot of sites and people haven't even measured organic carbon content. So they can't really, they're just guessing at the absorption. So if you know what the carbon content is of your different permeability zones in your system, you can at least assume linearity and get a decent absorption amount. So you can know how much storage capacity there is if you have concentration data from wells. At early time, the NAPL distribution dissolution processes will likely dominate your contaminant plume. And you can describe this using a linear driving force model, and this is pretty well established. At late time, contaminant release or back diffusion from low permeability absorptive materials can dominate plume longevity. And everyone's pretty much accepted this. But the key thing, and the part I struggle with sometimes, is what is meant by back diffusion. And to me, the two things that are incorporated that, there's diffusion and there's absorption, okay? So if you just think of a tracer, then you don't have to worry about absorption. But we have to deal with things that absorb. So it's both the diffusion and the absorption isotherm, how you represent that. And it may even be rate limited. So these can prolong contaminant tailing even longer than just diffusion. And so these are important irrespective of diffusional limitations to mass transfer. So my point here is be careful when you assume linear equilibrium absorption, because you may have tailing, you may not have any back diffusion, you may get tailing just because you have nonlinear, non-equilibrium absorption. All right, with that, I'm gonna hand it over to Natalie, and she's gonna focus on how biological processes influence the back diffusion.
Awesome. Thank you so much, Kurt. Hold on just a second. I'm gonna change the presenter here. Okay, Natalie, it should be getting passed to you.
Okay, 1 second here. Show my screen. All right, here we go.
Looks great.
All right, so that was a great intro provided by Kurt. Really gives everyone a better understanding of what are the mass transfer limitations and how, you know, NAPL and low permeability zones can potentially impact long-term monitoring of these sites. But now I want to focus on how the biological component impacts that mass transfer. So you may be familiar with NAPL, the NAPL water interface, and how the biological activity impacts that mass transfer. But I want to highlight the two key aspects that impact this. One, the biological activity can act to increase that driving force. So it lowers the bulk concentration, and as a result, you get biologically enhanced dissolution. So similar to what Kurt showed, now the biology is having an impact on this phenomenon. The other way that biology impacts mass transfer is by that transformation that we know biological processes to result in. So as PC and TC, say our parent compounds, transform those compounds to the less chlorinated compounds like DCE and vinyl chloride, the aqueous solubility increases. As a result, then we have more mass transfer. So just to refresh everyone's memory a little bit about the biology. So we have, again, we're starting with PC and TC, typically as our parent compound. And as we dechlorinate, we have an increase in solubility, hopefully getting down to non-toxic ethene. So this is an anaerobic process typically, and it requires an electron donor, and then the contaminant itself is the electron acceptor. Just like oxygen is for us, these microbes respire the contaminant. So the first couple steps of PCE, TCE to dichloroethene, DCE, can be carried out by multiple organisms, but then that process stops. One of the most well-studied organisms is Dehylicocoides mccartyi, and there are multiple strains of this organism that can carry out different steps, as you see, anywhere starting from PCE, going to vinyl chloride, and then hopefully we getting down to these organisms that are carrying out the transformation all the way to ethene. So there's genes associated. You see there's PCA, TCA, BVCA. These are genes that are associated with that function that allow us to identify which strains or group of or couple strains that are carrying out that process. So the two strains and the two genes that we're most interested in are the BVCA and the VCRA gene. And the reason for that is those are the two that are associated with ethene formation. So the studies I'll be talking about in just a second were carried out with the BDI-SZ consortium or the KB1 consortium provided by Serum. Both of these consortiums have multiple strains of the helicocoides. So we were able to identify the VCRA, the BVCA, and the TCA gene, and those are again associated with different strains of the helicocoides. So actually, I will be talking a little bit about combined remedies. First, I just wanted to highlight DNAPL dissolution, and this is in a system, another aquifer cell system. I've done a lot of these in collaboration with Kurt. So this was a system where we had injected our PCE, and then we flushed it with a tween 80 solution, removed 55% of the mass. And then Kurt talked about this ganglia to pool ratio. So at the end of that flushing with the tween 80 solution, we had about 50% ganglia and pools. And then we bioaugmented with a BDI, and then we provided lactate as their electron donor. And this work is published if you'd like to read more. There's a reference. So following that flushing, this is what the NAPL distribution, our source zone, looked like. You see the areas of ganglia up here, and we still have some pool remaining at the bottom. So now we've gone, this is starting with the biological process, the biological bioaugmentation. So bioaugmented about that point. See, in the beginning, we had all PCE here in blue. Within less than a pore volume, we had cis-DCE formation there in red. And then with about 10 pore volumes, we started to see vinyl chloride and ethene being formed. So Kurt talked about mass transfer limitations, and here's an example of that. These are 2 points in which we did a flow interruption. So we actually stopped our system. And during that time, immediately following those events, we saw big increases in vinyl chloride and ethene formation. So the microbes, while this system was flowing pretty slow, it was about 6 centimeters a day, they really benefit from having even more retention time, residence time. And at the bottom here, we see these pie charts. So it started out, all PCE. And then by the end of the experiment, once the microbes had really taken hold of the system, we had 60% ethene. So now looking specifically at the pooled and ganglia regions in our system, first, want to walk you through this lot of information here. So the bar charts here are our PCE to ethene. We want to see more red and pink and less gray. So the gray is the PCE. And then we also have plotted the organisms of, so I mentioned Geobacter, that's one of the organisms that takes PCE to cis-DCE, and then total to helicoides, so that's a summation of all the different strains, and then 3 specific strains that were measured based on their genes. And then this dashed line here is what we would anticipate the abiotic, if there was just abiotic dissolution alone, that's what we anticipate the PCE yield to be, the dissolution to be. So anything above that line on both of these plots is what we're calling biologically enhanced dissolution. So what we see is in the pooled zone, we have about 2.3 times more dissolution, biologically enhanced dissolution, and in the ganglia region, the greater surface area location, we have about 6 times fold biologically enhanced dissolution. This is over what you would anticipate with just abiotic, just water flushing alone. And then the VCRA gene, which is one of the organisms, which was one of the strains responsible for ethene formation, was primarily responsible for ethene in this location. And that becomes important as I show you the next couple slides. Okay, so we've seen, and many other people have seen, that biologically enhanced dissolution near DNAPL source zones can increase the rate of removal by up to 14 times. We're expecting a similar type of enhancement in terms of diffusion and desorption process in low permeability zones. And this is the abstract art from our paper. Up top, we have the abiotic system, we have a low permeability zone, and we just have PCE in there. And that slowly dissolves, back diffuses out of the lens. If the system is biologically active, we're going to have, again, those daughter products being formed, and we anticipate the organisms actually going into the lens and hopefully having an impact in increasing that diffusion out of the system. So despite, this analogous kind of connection between the dissolution and the diffusion, there hadn't been, as far as we could see, a study that had really looked at the fine scale heterogeneity and the biological impact on back diffusion. And then also, how do specific Dihelicocoides strains, again, associated with the formation of ethene, lead to that enhancement, and how can we better understand the biological processes that are going on? So the objectives of this work were to quantify that back diffusion, that biologically enhanced back diffusion, and look at the distribution of the Dihelicocoides. So very similar system. These systems are I forgot to highlight, this is a little bit smaller than the systems that Kurt showed, but this aquifer cell and the previous aquifer cell are actually the exact same system. Here, instead of having low permeability sands, we have 4 lenses with different hydraulic conductivities, different organic carbon content. Remember Kurt highlighted at the end there, it's really important to understand how organic carbon plays a role in terms of diffusion. So another thing we have here at the bottom is clay. It's actually clay that we've loaded with PCE. So that's obviously a very low permeability zone. And we looked, and then we monitored all these ports and looked at the biological Dihelicocoides abundance and the chlorinated ethene concentrations. And this work is also published and available. So there's a lot going on this slide. First, I'm just going to walk you through it. We started with a bromide and fluorescein tracer. So this was used to calibrate the mathematical model that we used. It was a pulse of bromide, and then we flushed it with water. And then we basically loaded up our system. We added TCE and then flushed it out. And this again was to help develop our model. And then we started the biological component. So we redeveloped our plume because pretty much all the TC had washed out during the abiotic portion. And then we reduced, and actually in both cases, we start with a higher concentration of TCE, and then we reduce it to monitor that back diffusion, as you would anticipate, like a source zone cleanup, up gradient. In the biological step, we also do the same type of thing, but we do a bioaugmentation step, and then we incrementally decrease the TCE concentrations, and we measure the Dihelicocoides over time. So first, the abiotic results. Again, we did the tracer, and here is the aquifer cell. This is the fluorescein tracer, and the bromide. So this was to develop or to measure parameters that were used to calibrate our mathematical model. And we found from the abiotic portion of this work was that TC, as you would expect, we can't quite remove all of it. Some of it is sorbed, as opposed to the bromide that almost was completely removed from our system. And as you would expect, there was more retention of TCE in the lower permeability and high organic carbon content locations. And here's an example. This is port 1E. This is right here at the bottom, close to the clay. The difference between the TC is in red and the bromide is in blue. You see that gap there. So that's mass that has been retained. And then in the inset there, that's at on the log scale. So now just a little bit about the model. This was a MT3D model, and what was done is we used the abiotic portion of our experiment to then predict what would happen under similar conditions, but not the exact same conditions, under our biological condition. But we turned off the biodegradation component. So we're comparing the biological measurements that we take versus the modeled abiotic concentrations. And that's how we then go about determining our biological enhancement. So these are our VOCs, our chlorinated ethenes and ethene, coming out of different locations in the aquifer cell. The locations are highlighted here in red. So we looked at down here at the bottom. This is port 1E, 2C, so just above that, and down gradient of a low permeability zone, and then another location also near a low permeability zone and near the clay, and then up here's the effluent. So we see transformation to vinyl chloride happen fairly quickly. after we lower that concentration. So these lines here, these vertical lines designate when we lower the concentration from, I believe, 0.5 milligrams, millimolar to 0.4 to then 0.01, excuse me, 0.04 to 0.01. And in all cases, I forgot to mention, you see that cis-TCE being removed really quickly, transformed really quickly, and the green is the cis-TCE. Sorry, TCE is being transformed to cis-TCE rather quickly, and then vinyl chloride and ethene are being formed. And there was sustained ethene formation much higher, about 2 to 4 times higher than the influent TCE at late times. So that's indicative that there was diffusion desorption from those low permeability zones, and as you would expect, that was highest near the clay. So now looking specifically at the biological enhancement. In the red here, we have the modeled values, and then in the blue, those are the measured values. And I would say the modeled values and the measured values closely match each other in terms of first trends, but there is a distinct difference. So the higher concentrations of chlorinated ethenes were measured obviously due to biological enhancement of the back diffusion and desorption from the low permeability zones compared to what would have been anticipated under abiotic conditions. And in the zones in green here is where we had the most enhancement. The numbers in parentheses are the cumulative over the entire experiment, and the numbers outside of the parentheses are since our concentrations were decreased, so since about 3 pore volumes on. And then down gradient, we had less biological enhancement. We did still have biological enhancement. Overall, there was about a 6 to 53% enhancement of the mass removal from these specific zones. Again, this is the clay where we had the most enhancement, and then down gradient of these low permeability zones and higher organic carbon locations, we also saw higher numbers of enhancement. So now looking specifically at the organisms, so throughout the system, this is total Dihalococoides, all the strains lumped together. There were minor differences throughout the domain, but there were slight increases, as you anticipate, just down gradient or near the low permeability zones, where there was still contaminant available, or basically a food source for the organisms to use. On average, the organisms grew about 2 orders of magnitude. We saw there was no decrease in the Dihalococoides even after the concentrations of TCE were dropped significantly in these areas where we had higher biological enhancement, and that's because there was contaminant still available in those zones for the microbes to use. And then up here where we had less enhancement, we did see a decrease of Dihalococoides abundance by the end of the experiment. But on average, all the microbes grew throughout the domain. Now looking at specific strains, also a lot going on here. So this is the measuring of aqueous concentrations of Dihalococoides, different strains at different points in time. So starting at day 2, 8, 16, and 37. So TCEA gene, which is responsible for the first few steps to vinyl of TCE to vinyl chloride, that was detected as you'd expect where TCE was still present. The BVCA and VCR gene, which are associated with ethene formation, were present in locations where cis-TCE was likely formed. And then the VCRA gene was located, or strains carrying the VCRA gene were located throughout the domain. And you really see as the concentration has been decreased, they start to take over, and that explains why we saw so much vinyl chloride and ethene towards the end of the work, towards the end of the experiment. So we always take aqueous concentrations for our Dihalococoides, and this would be our attached concentrations. Those would be numbers you would get if you took a core in the field site, at your field site. So what we found interesting was we actually took samples, so we destructed our system, and we took samples within the lenses themselves. And we found that organisms are actually able to penetrate different low permeability zones, including the clay. And they were present on the edges as well, which is really encouraging because that's where you want them to be living. So to increase the dissolution out of those zones. So TCA, which is again responsible for the transformation of TCE to vinyl chloride, that was primarily located on the edges of these lenses and just down gradient. The BVCA gene was primarily located where, again, electron acceptors were available, and those really within the clay, there's quite a few organisms harboring the BVCA gene, as opposed to, and what I found interesting is that the BVCA was located in zones where the organic carbon was a little bit lower, while the VCRA gene was also present where there would be electron acceptor available, but they really got a hold of the location, really grew in locations where organic carbon was highest. And we've seen this with other systems. The BVCA seems to fill in and take the place of those organisms carrying VCRA when it's not the most hospitable situation. And so they kind of, they act as cleanup. They're the cleanup hitters and result in ethene formation. So wrapping up, so the take-home messages here is we've seen that dechlorinating microbes can enhance mass transfer of chlorinated ethenes from low permeability zones. Cumulative, coming out of our effluent, we had about 12% enhancement. Locally, we saw up to 53% biological enhancement. And this is really important if you're considering M&A at your site, and we want to make sure you're incorporating these considerations and looking at whether system for one is amendable to mandatory national attenuation, but take into account that biological activity could potentially enhance the cleanup and enhance your cleanup and reduce the cleanup time. So the helicocoated cells were capable of penetrating these low permeability zones, including the clays. The distribution of organisms, as you'd expect, as indicated by the reductive dehalogenase genes, had the ability to access electron acceptors that were on the fringes of those low permeability zones, and their distribution was really a function of the physical properties of the material. So it's really, and certain organisms had certain preferences of where they were able to be most active. So it's really important to maintain a diverse community to achieve all the benefits of bioremediation. And then finally, the different species, different strains will grow in conditions, again, that are most favorable to them. So I want to wrap up with some acknowledgements. Frank Loeffler helped us with that first project I was talking about with the NAPL biological enhanced NAPL dissolution. And then Linda Abriola at Tufts, Jason Natko, and Laurent Yang, two newly minted PhDs. This was part of their PhD work. Jason did the experimental work and Laurent did all the modeling for the biological enhancement from the back diffusion from these low permeability zones. So that's their work. And also I want to acknowledge CERTA for the funding and Serum for providing the organisms for the last study. So with that, Kurt and I would be happy to take some questions.
All right. Thank you, Natalie. Thank you, Kurt, for the great presentation. We do have several questions. Again, if you have questions in the audience, you can enter them into your GoToWebinar panel, and we'll get through as many of these in the next 10 minutes as we can. So the first question up is, how do you measure the interfacial area in the field?
I'll let Kurt take that.
All right. That's a very good question. So to actually measure it, you can use what's called interfacial tracers. These are surfactant-like molecules that go to that NAPL water interface, and based on their retardation relative to a non-reactive tracer, a tracer does not interact with that interface, you can back calculate the interfacial area. This is a little bit complicated to do because some of those tracers will also absorb organic matter, so you have to take that into account also. It's relatively effective. You can do it relatively easily in the lab. The other way to estimate that interfacial area is to use data from lab studies and the type of porous media. You can estimate interfacial areas based on work that's previously been done in labs. So it's not a trivial measurement for sure.
I think this one may be for you as well, Kurt. How large were the test cells? And it says it looked like an initial slide. There were measurement in centimeters, and they're wondering how the findings mimic real situations, and if researchers have found correlations between that lab model.
Yeah, that's a good question. So some of the cells we have generally range. The largest ones are about 50 centimeters high and 1.5 meters long. But some of the other ones are about 40 centimeters tall and about 60 centimeters long. And so generally, the largest ones we've used are 1.5 meters in length. So that's about the scale you're looking at. We recently did a field study at a site in Vermont where one of the objectives was to look at how the data that myself and Natalie obtained in a 1 meter long scale could be translated to the field site. And we didn't really present that today, but the idea was that if you took a grid block of a field scale model was like 1 meter, we're trying to represent that in the laboratory and then translate that, upscale that to a mathematical model into a field site. And it actually worked pretty well at those scales. So the field site we worked at was on the order of 10 meters. And so we had basically a 1 meter simulated system that was able to upscale the parameters we measured to that field site and then reasonably predict the results. I think Laurent is still working on that paper, but it should be coming out soon.
Yeah, it's submitted. I think Linda just got comments back on it. So stay tuned for that. Yeah, we looked at rates between microcosm, these aquifer cells, and then our pilot scale field study and compared those. And if I remember correctly, is taking into account heterogeneity played a huge role. So doing a lumped parameters over the domain was really where you saw the biggest error.
Okay, the next question is, when you're setting these up, do you use saturated soil samples to look at the contaminated areas, or are you doing groundwater only? And if you do look at saturated soils, how do you do the mass calculations? Are you measuring TOC or other parameters?
I'm not sure I exactly understand the question, but if you try to do the phase distribution, you have to measure the organic carbon content of the soil. So you have to know those parameters. You know the aqueous phase concentration, and you know the total concentration. And so from that, you can back out the phase distribution. I think that's what the question was about, but I'm not positive.
I think that's what they were asking. If it's not, if you can submit something else, we can make sure we clarify. The next question is, have you looked at this for petroleum-based NAPL products as well?
I did some early experiments with dodecane, which was used in some of the jet fuels, and doing some work now with JP4 jet fuel. But in general, we were primarily focused on, or at least I was, mostly on chlorinated solvents. Natalie may have another answer for that.
Yeah, I mean, I did a little bit of work with ethanol and BTEX, but not to this extent. It was more looking at I'd say the dissolved fate of dissolved fate compounds and the biotransformation, but nothing with different phases, nothing in the NAPL phase.
I would say that like the physical chemical processes are, I think they're translatable to different compounds as long as you know the physical properties and absorption behavior. And similarly, I think if you understood the degradation as well as what Natalie presented, you could also look at how that microbial activity impacted mass transfer.
Okay, Okay, there's a couple of questions about some dihelicocoides and kind of what you guys have seen in these studies that are very similar. How often have you seen the dihelicocoides penetrate into the low permeability layers, such as clays? And are you seeing, low K layers have different rates, and how predictable is that?
So yeah, we did see the organisms penetrate into all the different zones. I don't know if my slide is still visible right now, but the organisms did get into the clay zones. They got into different organic carbon content locations, the appling soil, the web. They got into all these lenses. What we found was different strains, dihelicocoides strains, were better able to penetrate the locations, such as the clay, than others. As I was saying, I think the BVCA, the strains carrying the BVCA gene were better able to penetrate the clay than some of the other organisms.
All right, this question is for you, Natalie. Is there any published evidence that you've seen that dihelicocoides can produce biosurfactants or other mass transfer enhancing molecules?
Not that I'm aware of. I would say somewhat related, but they definitely do attach and form biofilm. I don't know about a biosurfactant though. And I would say translatable to the field, it's really important to look at both the attached and the unattached organisms, because we found that the attached organisms are often those that are active. So that's not really active. I don't know much about the biosurfactant part in terms of the dihelicocoides.
Yeah, I haven't seen anything published on that either. As far as the abiotic degradation of CVOCs, did you guys look at this, especially in those low K zones?
We didn't specifically. I mean, we did monitor. I guess we didn't have any unexplained ethene formation. So that's one indication, maybe that we didn't have any abiotic transformation. But we didn't check for acetylene in this study. I did in the first aquifer cell that I showed. We did monitor for that, and even though I did have iron sulfide formation, I didn't have any indications of abiotic transformation occurring. We looked at ethene, ethane, and acetylene, and we did not detect any of those. So I can't comment on it for the aquifer cell with the low permeability zones, but for the first aquifer cell I showed with the NAPL, the PC NAPL combined with the surfactant, we did check for it, but we didn't find any evidence of abiotic activity. Abiotic.
Great. All right. One final question on this, because I know we're out of time. Have you attempted this type of work with any of the perfluorinated compounds? Looking at, I think that's exact. They just kind of repeated it, but anything with perfluorinated that's been done on that?
So that's kind of something we're working on now. So Kurt and I are, along with Brenda, working on a project looking at fate and transport of PFAS compounds, looking at the precursors. Kurt's doing the fate and transport, and I'm doing the biological part. We haven't done any systems with flow, with abiotic, or looking at absorption and then the biological part yet. So hopefully soon, I'm not soon, been a year or so.
Hopefully we'll have a follow-up showing the results from that work.
Yeah, I hope we have some results that are positive.
All right. Well, thank you everyone for joining. Thank you, Kurt and Natalie, for this amazing presentation. We're really excited about that. If you have additional questions, you can reach out to Kurt or Natalie directly, or you can send them to info@microbe.com, and we'll be happy to answer any of those questions for you. I think Natalie's, can you go to the slide, Natalie, that has your email addresses, just in case anyone wants to reach out? And we'll post those up for you guys. And the recording, too, will be on the resource section of the Microbial Insights website, so you'll be able to watch this excellent presentation again and kind of think through if you have additional questions. But again, thank you very much, Kurt. Thank you, Natalie, for this wonderful presentation. We appreciate your expertise and sharing your research with us.
Thank you.
Thank you, Nora. Thank you, everyone.
All right. Thank you, everyone. Have a great day.